we are going to talk gene regulation. So gene regulation builds on the idea from year one biology that you have chromosomes, and the chromosomes, homologous pairs of chromosomes in the corresponding positions have the same genes, and different genes encode different or ultimately affect different characteristics. But how do they do that? Different homologous chromosomes may have different versions of genes called alleles, but each allele is ultimately, it's a stretch of the DNA, and the way that that gene affects a characteristic is by encoding a protein. So the DNA, section of that DNA then at some point will undergo unwinding and a section of that DNA, so the unwound section, will be copied or used as a template to make RNA, mRNA. The DNA will then wind back up again. The mRNA will then go into the cytoplasm, where a ribosome will bind to the mRNA, and the ribosome will move along the mRNA, and according to each set of 3 bases that it reads on the mRNA, it will accordingly use transfer RNAs to add specific amino acids together, join them together, forming peptide bonds and connecting amino acids in the order that is determined by the sequence of codons on the mRNA. So amino acids will be put together as the ribosome moves down, and ultimately we will get a polypeptide chain. Shall we do that in a different color? Ultimately we will get a polypeptide chain based on the sequence of codons in the mRNA, and the sequence of codons in the mRNA determined by the sequence of triplets or bases in the DNA in the gene or in the allele. So our polypeptide chain then undergoes folding. Remember that polypeptide chain's got a primary structure, specific sequence of amino acids, and that specific sequence of amino acids begins folding into alpha helices or beta pleated sheets according to whatever sequence of amino acids there are in the polypeptide chain. But once it's done that, it's going to adopt its tertiary structure, where based again on the sequence of amino acids, there's going to be interactions between the different R groups of the amino acids in that polypeptide chain. So the nature of the R groups and the positions they find themselves in the polypeptide chain is going to determine where these R group interactions take place. So remember, these are disulfide bonds, they are ionic bonds, they are hydrogen bonds, and there are also hydrophobic interactions or non-polar interactions, or Van der Waals forces, whatever you want to call it. Okay, so you got these, and that determines the tertiary structure, and the tertiary structure ultimately determines the function. Okay, and proteins, they can be enzymes, they can be transport proteins, they can be channels, they can be pumps, they can be antibodies, they can be structural. So it's essentially, the point being made is that your genes have control characteristics, and my point being here is that whatever the function of the protein is in the body, that is what determines characteristics. You can call it phenotype. In summary, the first essential concept that we need to remind ourselves from year one is of gene expression genes are expressed mostly through the production of proteins. And because it's the proteins that actually do things in the body, and it's the doing things in the body that ultimately gives the organism its characteristic features, functions, structure, and so on. Okay? But we knew that in year one, or we should have, that this is how genes work. Now in year two, the important concept is of gene regulation. Okay? So somehow we need to be able to control this process a little bit more than we see here. So the next thing that I will discuss is what gene regulation is and why we need to do it. So point 2 is going to be gene regulation. Okay. Now, you know, in previous years, I have focused a lot on the mechanisms, details, and workings of things. But the more I do this, the more I realize that actually it's when the big picture makes sense that the rest of it falls into place a little bit more quickly, a little bit more easily. It's easier to remember details when you know how the details work together in the big picture. So I'm going top down. So we've discussed gene expression, and the next thing we're going to talk about is gene regulation. And in my view, gene regulation comprises, or let's first talk about why we need to do gene regulation. Why is it even a thing? So gene regulation, why is this? That in many cases, you need genes to be expressed. So you want the protein to be made from the cell, from the genetic information in the nucleus. But it's just as important in organisms to be able to turn genes off and to prevent them from being expressed when they are not needed. We'll look at some examples. Okay? So, I mean, the most obvious example in terms of on and off in genes is, for example, when you have differentiation. Okay, and this kind of is following on to the idea of development of organisms and body plans. Okay, but the idea of differentiation, let's even call it differentiation / development, is the idea that, and this is the bigger idea of gene regulation, is that in an organism, within an organism, you have different tissues, different cell types, and so on. But all those cells have the same genetic information. They have the same genes and alleles. So how is it that a stem cell can differentiate into different types of cells? Okay. So we've got this stem cell can differentiate into this type of cell. It can differentiate into this type of cell, and it can differentiate into this cell too. Okay. How is it able to do that? And the idea is that cells, in order to become differentiated, or in order to change, cells can turn genes on and genes off. So let's say this stem cell, we're talking very basic organism, it has in its genome, it has genes A, B, C, D, and E, all on its chromosomes. Okay? In order to become a muscle cell, so in order to become this cell type, actually let's call it 1, 2, 3, 4, 5, 6. Okay? If you want to be a muscle cell, you need to express genes A and B but genes C, D, E, and F must be off, because they're the genes to become other things. So in order for this cell, in order for the stem cell to differentiate into a muscle cell, for example, genes A and B need to be on, but genes C, D, E, and F must be off. Now you can't delete that DNA, okay? But what you can do is not express the genes. So, you know, we'll be transcribing and translating the information from genes A and B, and we'll be making proteins from gene A and B, but we won't be making proteins from genes C, D, E, and F. And similarly, in this neuron, for example, we will have A and B off, but we will express C and D, and again, we will not be expressing E and F. We need to repress those genes. We will not be expressing those genes, but expression of genes C and D will cause this cell to express the right proteins for it to behave as a neuron. And by extension then, okay, just carrying on, A, B off, C, D off, and E and F expressed, used to make proteins. And because each of these cells have different proteins doing different things, having different functions, these cells then start behaving differently and are differentiated and, you know, carry out different specialized roles that collectively help the organism to function and survive. So, you know, from that point of view then, I hope we can see why being able to turn genes on and off is important, because cells can't delete the DNA that they don't need. Okay, so how are they going to behave differently? By being able to turn genes on and off. Let's look at another example. So that's the on-off idea. But the other situation where you need to regulate genes is this idea that it goes beyond on and off. So even in the on situation, so even when genes are expressed, there might be scenarios where you need less, or in different scenarios you might need more. Okay? So while on and off, you know, in my view, is more to do with differentiation, and it might have more to do with development. So this less and more, and what we're talking about here is down regulation. So something's expressed, but we just need less of it right now. And more is to do with up regulation. So it's expressed, but all of a sudden something's happened and we need more of it. This, I feel, is more to do with signaling and responding, as we kind of seen in other parts of the course. Organisms, remember, organisms have sensors. They respond to stimuli from the environment. They have communication networks, the endocrine system or the nervous system, and these communicate to effectors, and the effectors make a response. Now, one of the responses that cells can make is to alter their gene expression. So in this story, you know, being able to make less of something or produce more of a protein, it might be a response to a signal. So some homeostatic mechanism, some external stimulus has happened, and cells can respond by altering their gene expression, altering the expression of certain genes that will kind of make a response. So I hope you can see that not only do we need to be able to switch genes on and off, but we might also need to upregulate genes and downregulate genes that are already in an on situation. Okay, so now that we've discussed the bigger concept here, and remember, it's the concepts that's going to help you to apply things in exams. So in an exam, they might give you a situation where you know, explain how stem cells can be used to form heart or cardiac tissue. Now, in that case, you'll need to think about the bigger idea, about what part of the content that's referring to, and then once you've made your decision on that point, then you can detail how that mechanism could work, but that's when your details come in. Okay, so now what we'll do is move on to how this is all happening, or how it could happen. So now let's talk mechanisms, and I think it might be good for us to just retrace the protein or the gene expression kind of route, okay? So we had DNA, okay, and the DNA is used to make RNA, transcription, and once we have the RNA is then used to make protein, and the process that does that is translation happening at the ribosome. Okay, and remember, our protein will eventually then have a function, and it's the function that we're interested in. This is the function that that's the reason why we are doing any gene regulation, is because we're interested in manipulating and changing that according to requirements. Okay, so all begins here, but it's this that we're interested in. So we call it gene regulation, but really we're regulating the function of proteins. Okay, right, so the reason I've done this is that the gene regulatory mechanisms take place at all of these stages. Okay, so there's mechanisms that regulate DNA, there's mechanisms that regulate transcription, there's mechanisms that affect the RNA that's produced from the transcription, there's mechanisms that affect translation, there's even stuff that's done to the protein, all with the view to changing and modifying this. All right, so make sure you get that, and now we'll get into it. So the first place that genes are regulated is in the structure of the DNA itself. So DNA can be, so we need to remember that DNA in the year one, we might have just thought of it as a double helix, two strands of polynucleotides, hydrogen bonded together through complementary base pairing, and forced into coiling because of interactions with water. Okay, however, during cell division, the DNA gets super coiled and forms the structures we can see down the microscope as chromosomes. So DNA can become very condensed, and it's actually the case that DNA is usually in the structure called chromatin, where the DNA strand is wrapped around these proteins called histones. Okay, so this is a histone protein, right? That's your DNA double helix, right? And you probably got various genes along the stretch of this. Okay, this structure is called a nucleosome. The DNA wrapped around the histone protein. Okay, and it turns out that, so this is how DNA usually spends its time, in the cell. Now, when the cell is dividing, the DNA becomes very condensed, like this. Okay, and in that case, it's really wrapped up very tightly, so that there's hardly any loose DNA. Okay, so you got twice wrapping. So when DNA is very condensed, it is like this, and in this form it is called euchromatin. However, when it's not undergoing replication, or when the cell is not undergoing replication, DNA tends to be a bit more loosely wrapped around the histones, and it looks a little bit more like this. Okay, and so there's these free regions, there's a little bit of loose DNA between the parts that are wrapped around the histones, and this is called heterochromatin. Heterochromatin. Now, the key point being this, is that when DNA is in this form, right, when DNA is in the euchromatin form, then there is little access to RNA polymerase. So there's so little free DNA that RNA polymerase cannot access any of the genes. Okay, little access to RNA polymerase, so genes not accessible for transcription to occur. So transcription can't occur when DNA is so tightly wrapped around the histones and there's no free available for RNA polymerase to bind to. However, when DNA is in this form, RNA polymerase can access. So RNA polymerase can access genes transcription can occur. All right, so that's the key point here, that you can have these two forms of DNA, one in which gene expression is restricted because RNA polymerase can't access the DNA in order to bind it and transcribe the gene, and another form where genes are accessible, RNA polymerase can bind to those genes and transcribe them, and therefore we can get protein production. It so happens that there's certain modifications that you can make to specific genes to make them in this form, whereas other genes you can keep them in this form. So we're envisaging a situation where you can have like some DNA in a cell. So let's just say we're looking at one chromosome in a cell. We want to express these genes, so we're keeping them kind of in the heterochromatin form, but we want these genes switched off, so we're supercoiling them, or we're coiling them, or we're having them in the euchromatin form, tightly wrapped around the histones, DNA not accessible to the RNA polymerase, and other genes we're having them on, other genes off, other genes on, other genes off. Okay, now what controls that? What controls the form in which certain sections of DNA is going to be? So there are enzymes which can be regulated by other signaling processes happening in the cell, okay, that can do certain modifications. So if you add acetyl groups, so acetylation, and these are all modifications that are happening to the histone proteins. Other modifications are also possible directly to the DNA molecule, but we're going to be talking about histone modification. So, the way that you control which form DNA is in is by modifying the histone proteins. So enzymes can be directed to carry out acetylation of histone proteins, adding acetyl groups to them, or we can phosphorylate the histone proteins, and acetylation and phosphorylation results in DNA that is more loosely bound in a form that is making DNA accessible to the RNA polymerase. In the opposite way, histones can be, so let's just call these plus modifications, but the negative modification is that the histone proteins can be methylated. Now, when methyl groups are added to the histone proteins, then DNA wraps more tightly around them, and it results in the DNA tending to be more like euchromatin, where the DNA is wrapped around the histone proteins and not accessible, and we get less expression of those genes. Okay, so I hope that is making sense, right? And so now let's move on to the next. Well, actually, before we say that, what we should kind of mention is that what this will do is it will reduce transcription, or this is what will affect transcription. Okay, so whether DNA is in, so if the DNA is in this form, we're going to get less transcription and therefore less mRNA production and therefore less protein production, and therefore we're going to have less expression of the gene. However, if the DNA is in this form, we are going to get more transcription and therefore more translation and therefore more protein production and therefore more expression of that gene. So I hope that is clear. Right, next we will continue to talk about transcription, but we will talk about transcription factors and how even if DNA is accessible, we can still regulate the genes from that point also. Okay, so what we'll do is at the DNA stage, we'll just add, sorry, what's it called? Chromatin remodeling, isn't it? Okay, so we'll just put mechanism 1, chromatin remodeling. Okay, now we'll look at mechanism 2, still affecting the level of transcription happening, but it's using proteins or DNA binding proteins to regulate the transcription. Okay, now the example that's given is the Lac Operon. Do I want to go through the Lac Operon again? Is it the same? No, it's not the same. I will go through it. Okay. Right. So, okay. The general idea is this. I'm going to talk about the general idea, then we look at specifically Lac Operon. The general idea is this, that you got a gene, it's accessible. Okay, so what's going to happen? RNA polymerase is going to bind. Right? RNA polymerase binds and transcribes the gene, and we get mRNA. Yay! Okay, but it's not as simple as that, because RNA polymerase doesn't just randomly bind any exposed gene, at least not in a significant way. It usually takes a bunch of other proteins to already be there, and they are what we call transcription factors. So transcription factors are there, they bind the DNA first, and if they are there, then RNA polymerase binds in a significant way. Okay, RNA polymerase then binds because the transcription factors are there. So technically what we say is the transcription factor proteins, or the DNA binding proteins, they recruit RNA polymerase, because RNA polymerase isn't just going to turn up, right? So these transcription factors are needed first, they recruit RNA polymerase, and when RNA polymerase has been recruited, only then will it transcribe the gene and produce for us mRNA. Yeah, so that's great. Okay, these proteins are acting in a positive way. However, transcription factors, like nature doesn't miss an opportunity. Okay, so transcription factors, as well as being positive regulators, they could just as easily, other proteins can be negative regulators. And so when they are bound, when these proteins are bound, they actually prevent RNA. Why did I suddenly switch to green? they actually prevent RNA polymerase from binding. So those transcription factors, or those proteins there, maybe we don't have to call them transcription factors anymore. They can be just repressors of some kind. But these proteins, they bind the DNA, but instead of recruiting RNA polymerase, they prevent it from binding, and thus, again, we've regulated the gene expression, except this time in a negative way. So we've got less mRNA production. So this concept is important for us to understand when we look at the Lac Operon, because it kind of utilizes both of these mechanisms in a way. Okay, and you know, the only reason, or the reason why understanding the Lac Operon is important is it's kind of an example of how other gene regulatory mechanisms are taking place, not only in bacteria, but if bacteria have it, then all the organisms that are, you know, more further or highly evolved than bacteria. Okay, so right, so the Lac Operon is an example of how you can control or regulate transcription, and it kind of comes back to our idea of responding to your environment. Okay, so the Lac Operon has a certain, well, let's do this a bit bigger. So this is a bacterial cell, and we've got our loop of DNA, and we're not too worried about the genes on the other side of the loop, but right now we're looking at these genes right here on this side, and these genes quite handy because they allow the bacteria to utilize substrates other than glucose when glucose is not around. Okay, so there's a few genes in the Lac Operon. I don't think I'll refer to all of them. So this is a stretch of DNA, okay, base sequences, nucleotides, all that business. But then what we have a promoter region, we have an operator region, and then we've got the Lac genes. So we've got, I can't remember what they're called now. Also we have Lac Y, and we have Lac Z. Okay. Now, what we're saying is that most of the time, these bacteria, they have glucose available. Let's imagine. And glucose is their preferred respiratory substrate. So glucose enters the cell, glucose then goes through the respiratory metabolic pathways, and then we end up with CO2, but then the bacteria is able to use those reactions to convert ADP to ATP, thereby providing the ATP for what the bacteria needs the ATP for, and for whatever energy requirements the bacteria has. So that's the process. However, what we are saying is that sometimes the glucose is not available, and that other substrates are available, and by other substrates, the example that we're going to use is lactose, right? Now, lactose is a disaccharide, and in order for the bacteria to use the lactose, we'll do this later. So lactose is a disaccharide composed of glucose and galactose. So in order for the bacteria to use the lactose, they need to express additional genes. Lac Y, the gene Lac Y encodes a protein that acts, so the Lac Y is actually the gene for a protein called lactose permease. Does exactly what it implies. It allows lactose to enter the bacterial cell. That's great. So now lactose can be inside the cell. The LacZ encodes a protein that is called beta-galactosidase. And what it does is it hydrolyzes lactose, which is a disaccharide. It hydrolyzes, it breaks the glycosidic bond by the process of hydrolysis and converts it to glucose and galactose. And once it's done that, the glucose that's produced from that can enter the respiratory pathway. I hope we can see that in order to use lactose as a respiratory substrate, we need to express Lac-Y and Lac-Z, otherwise called the structural genes. Okay? Now, but the thing is that it wastes a lot of energy and resources to keep expressing these genes when there's no lactose around and when glucose is still around. So I hope we can see that there's a need here to make sure that these genes are not expressed when glucose is around, but are expressed when glucose is not around and when lactose is available to be used. Okay, so we've got our situation set up. Now let's see how that happens. In a situation where, let's say, there is no lactose, but there's the preferred substrate glucose is available, what will be happening is one of the genes in the Lac Operon encodes a repressor protein. Okay, so we have a repressor protein, and the repressor protein binds to this operator region right here. And when the repressor is binding the operator, RNA polymerase cannot bind to the promoter. So when the repressor is binding to the operator, and because it's taking up that space, it prevents RNA polymerase. RNA polymerase would normally have bound to the promoter. Okay, and when it would bind the promoter, it would be able to transcribe the genes in this operon. Okay, the structural genes Lac-Y and Lac-Z, and we have lactose permease, and we'd have beta-galactosidase, and we'd be able to use lactose, but we don't need them right now. So the repressor binds to the operator, preventing RNA polymerase from binding. So RNA polymerase is prevented from binding the promoter and thereby transcribing the genes. Okay, now when lactose is around, what happens is that some lactose is able to come in because genes, the way I have it in my head is that the gene isn't completely always off. Okay, so we might have some lactose permease still in the cell, or still in the cell membrane, that will allow some lactose to come in. When that lactose comes in, it binds to the repressor, and when this happens, it changes the shape of the repressor slightly so that the repressor can now no longer bind to the operator part of the Lac Operon. Okay, what that means is that RNA polymerase can now bind to the promoter, and it can now transcribe the genes, the structural genes for Lac-Y and Lac-Z, and we can get lactose permease expression and beta-galactosidase expression, and we can start to use lactose a bit. But remember what we said, that RNA polymerase isn't just going to bind here. In order for this expression to be at a high level, we need transcription factors, we need other proteins to recruit RNA polymerase more frequently to the DNA. Otherwise, we're just relying on kind of RNA polymerase randomly bumping into the exact spot that it's supposed to be in. Okay, so we need other proteins, and And that kind of makes sense because while glucose is still around, we don't really want to invest too much energy and resources into synthesizing these genes because we don't really need them yet. Yes, lactose is there, but glucose is still there. Okay? So how do we manage that? How do we make sure that while glucose is still around, we don't get that much expression of the Lac Operon? Okay? And this is where our transcription factor or activator protein comes in. So there is another protein involved in this story called the cyclic AMP receptor protein, okay? Otherwise called catabolite activator protein, and I'm pretty sure that it has other names too, okay? So let's just call it CRP for now. Cyclic AMP receptor protein. Now this protein, what it would do is it binds cyclic AMP. We should remember cyclic AMP as a modified nucleotide from our previous work on cell signaling, second messenger, stuff like that. Okay, so the cyclic AMP receptor protein would bind cyclic AMP, which would allow it to bind the DNA here to the Lac Operon. Right, so the CRP binds cyclic AMP, and when it does that, it binds here, cyclic AMP attached, and when it binds, it actually acts as a recruiter for RNA polymerase. So it helps RNA polymerase to bind to the promoter in the Lac Operon and to increase the transcription of the Lac Operon structural genes. Problem is this: when glucose is still around, the cyclic AMP levels in the cell are low. So when glucose is around, the cyclic AMP levels are low, which means this is not happening. The CRP does not have cyclic AMP available to bind to, and therefore it does not bind to the DNA, and therefore it does not recruit RNA polymerase. Which is good, which is what we need, because glucose is still around, and we don't want to transcribe these genes because our preferred substrate of glucose is still there. We don't need to use lactose. Now, let's imagine the situation where glucose has now become scarce. Glucose is no longer around, but lactose is. Only in this situation do we really need to express the lactose metabolism genes, the Lac Operon structural genes. Okay? When glucose isn't around, then cyclic AMP levels begin to go up. When cyclic AMP levels go up, then cyclic AMP is more likely to bind our cyclic AMP receptor protein. And when those two associate, the cyclic AMP receptor protein can bind the DNA, it can recruit RNA polymerase, and we do get high levels of transcription of the lactose metabolism structural genes. Okay, so when that happens, we get plenty of production of lactose permease, so lots of lactose can enter, right? And then we get lots of production of the beta-galactosidase enzyme, so we get lots of conversion of lactose to glucose, right? And then, because of that, we've got plenty of respiratory substrate so that, to synthesize our ATP with. Yeah, so I hope we can see where that's an example of we're kind of responding to our environment. It's an example of switching a gene on and off, and it's also an example of upregulating a gene that is technically already on but significantly upregulating it when the situation or when the need arises. Okay, so that's another mechanism by which we have controlled transcription, the idea of DNA binding proteins, the idea that these DNA binding proteins can either recruit RNA polymerase and increase transcription, or block RNA polymerase from binding the DNA and thereby reducing transcription. Right, and these DNA binding proteins themselves are controlled by various other mechanisms. It could be cell signaling mechanisms, it could be binding to molecules, it could be hormone signaling. These other proteins are themselves controlled to act as either active repressors or inactive repressors, or active transcription factors or inactive transcription factors. Right? So we had chromatin remodeling, and two, still in the area of transcription, we had, I'm going to just call them generally, DNA binding proteins. DNA binding proteins, and I will add also transcription factors and repressors. Okay, our next level of control of this process is going to be at the RNA. So we've at the RNA stage, i.e. we've got DNA, we found an accessible gene, we transcribed it because no repressor got in the way and we had enough transcription activating proteins present, and now we've got our RNA. Now, even at this stage, changes happen to the RNA that's produced that affect the protein that is made. When the DNA is transcribed, so we've just made a complementary copy of a section of the DNA, that initial mRNA molecule is called pre-mRNA for reasons that we'll see. So it's not the final, it's not the molecule that's eventually going to leave, or that will leave the nucleus yet. There are changes that happen to that molecule. Okay, so the first thing that happens to the pre-mRNA is that bits are added to both ends of it, and we'll see what effect this has. So the first two things are, we add to the 5' end, we add a 5' cap, and to the other end of the molecule we add a poly-A tail, just a long series of A nucleotides. So that's the first thing. And the second thing is that the pre-mRNA molecule is pretty much an exact copy of the DNA sequence, and the DNA sequence contained both non-coding regions and coding regions. So we had coding regions and non-coding regions. The coding regions are called exons, and the non-coding regions are called introns. Now, the reason for their existence will make sense later on. But for now, what we need to mention is that the pre-mRNA, because it's an exact copy of the DNA, it also contains the introns and the exons. And what we need to do is remove all the introns, because as they are non-coding, the information in those sequence of bases, we do not want that to be in the final mRNA, because only when we join the exons together that we get the right sequence of codons that is going to form the correct primary structure in the polypeptide, and therefore the right shaped protein that can work. So in order to get that, we need to remove the introns. So this is done in a process called splicing. So what we say is that the introns are spliced out. So all the introns are cut out and the exons, the remaining exons, are joined together. So you get a shorter molecule, still got the 5' cap, still got the poly A tail, but by this time we've only got the exons. So 1, 2, 3, 4. Had 4 exons. So exon 1 joined to exon 2, joined to exon 3, joined to exon 4, and that is our final mRNA molecule, and that will be leaving the nucleus and binding a ribosome and allowing the process of translation to occur. Now, how does this equate to regulation? So the way I see it, there's 2 main points at which regulation can occur here. Right, so the first point at which regulation occurs, or could be considered to be occurring, is the addition of the 5' cap and the poly A tail. These two, so first, the 5' cap and the poly A tail, they increase the stability of the mRNA. So we have to imagine a situation where mRNA in the cytoplasm is constantly being broken down as we don't need certain genes to be expressed. Now, the 5' cap and the poly A tail protect the mRNA from such degradation for some time, and so the overall effect that you would have is that the mRNA hangs around in the cytoplasm for longer, therefore increasing the chance that it's going to be used for translation, thereby increasing the amount of protein that can be produced. Okay, so the effect of increasing the stability of the mRNA is that we get more protein produced by translation. More protein produced, and therefore we have increased expression. We have increased expression. So I hope that makes sense. The other thing is the splicing. So if we've joined these exons together, so if I number the exons, so we had exon 1, exon 2, exon 3, and exon 4. Now they got joined together, exon 1 to 2 to 3 to 4. And that produced one mRNA. mRNA 1. Great. But at this splicing stage, what could also happen is something called alternative splicing. And what that means is you can regulate this process so that you can join the exons back up in a different order to what they are in the original DNA. So for example, at this stage, just as we've produced this mRNA number 1 with exon 1 joined to 2, joined to 3, joined to 4, at this point we could have equally joined exon 1 to exon 3 to exon 4 to exon 2. Now that produces a different sequence of codons in the final mRNA, so technically that's a different mRNA. That's mRNA 2, and we will get, because the sequence of codons in both of these mRNAs is different, we will get different proteins from each one. So from this one we'll get protein 1, and from this one we'll get protein 2. And because of that, we have therefore affected the protein produced from the gene, and therefore in a way, we've changed the expression of that gene. We've changed how the genetic information has been expressed by changing the sequence of codons in the mRNA and producing polypeptides with different amino acid sequences, therefore different tertiary structures, therefore different functions. So point 2 is alternative splicing. So point 1 was we're increasing the stability of the mRNA, but point 2 is we can produce different proteins from the same pre-mRNA molecule. So point 3 will be that we can affect the mRNA, actually we'll just call it RNA, we can affect the RNA processing. Okay, and in that RNA processing there were 2 main points: the addition of the 5' cap poly A tail that affected the stability of the mRNA. As well, we carried out splicing, removing the introns, but also having the ability to swap the exons around and produce different proteins. So the next mechanism was alternative splicing. So those are taking place at the RNA stage. Once the RNA has been produced by transcription, stuff can still happen to it. Okay, so now the next thing is going to be, once we've got our mRNAs, we can still affect how often they are used for translation by the ribosome. In our journey, we've produced some pre-mRNA, we've made some modifications to it, possibly switched some exons around, and because of that we now have a much smaller molecule. It's got some modifications on it, but now it is exiting the nucleus via the nuclear pores, coming into the cytoplasm. Now in the cytoplasm, the mRNA can still be affected in terms of how often it's going to meet a ribosome to carry out, to allow the process of translation to occur. Okay, so remember that the 2 parts of the ribosome will attach to the mRNA. It will move along the mRNA, and as it does so, tRNAs will be bringing amino acids according to the sequence of codons, connecting them up. The ribosome will be connecting them up in the order determined by the sequence of codons here, and producing the polypeptide, which will become the protein. But there are things that affect the binding of the ribosome to the mRNA. Okay, so just like we had transcription factors that controlled how often RNA polymerase was binding the DNA, we have these proteins that affect the binding of the ribosome to the mRNA. All right, so we can call them mRNA binding proteins, but they're like proteins that associate with the RNA and either help or increase the binding of ribosomes to the mRNA, or decrease the binding of the ribosome to the mRNA. Now what will happen is that if these proteins are acting in a positive way, then the ribosome will be recruited to, or the mRNA binding to the ribosome will be increased, and we will get more expression of the protein because it's being translated more often. Or these proteins could be acting in a negative way and preventing the ribosome from binding the mRNA, and therefore even though the mRNA is there, we might get less translation of the mRNA, and therefore less protein production, and therefore less gene expression. Okay, so that's the point there, that even at the stage of translation, these mRNA binding proteins can act in a positive or negative way. Again, signaling events in the cell will be controlling which proteins are binding to that mRNA, as well as the sequence of the mRNA. Okay, so we'll add point 4 here. We'll add mRNA binding proteins. And we'll remember that they can act in a positive, so some proteins will be acting in a positive way, some proteins will be acting in a negative way to affect, so they latch onto the mRNA and they'll affect how often the mRNA will be used for translation by binding to a ribosome. Let's move on to protein now. So even after we've got our polypeptide, many things can happen to the protein to regulate its function. Many things. Okay, I'm just going to list a few here, but a lot are possible. So once you've got a protein, many things can occur. We have post-translational modifications. So one of the things that you can do is add lipid to that, modifying the function of the protein by adding lipid. You could add carbohydrate to the protein, as we've seen with many membrane proteins have carbohydrate attached in the Golgi apparatus. So you can do that. You can have, and this is one of the big ways that protein function is regulated in the cell, you can add phosphate groups. Okay? Now this changes the protein's shape and behavior. So some proteins are activated when you add a phosphate group to them, because remember, whatever you do, this is just adding a phosphate group, it's one of the things used by the cell, but adding a phosphate group, highly negatively charged, is going to affect the tertiary structure. Okay, now if you do that to a protein, you could activate it, so that change could activate the protein, or the addition of the phosphate group could induce a change that's going to make it less active. So adding phosphate groups or phosphorylation is one of the most common ways that protein function is regulated in cells. You can add phosphate groups, you can bind to other molecules or ions. So many proteins are regulated by, or their activity is modified by, binding other molecules. So we saw the repressor the repressor protein. It normally bound the DNA in the Lac Operon, but when lactose was bound to the repressor protein, its shape changed, and now it did not bind, or it does not bind the DNA. And many proteins work like that. So, for example, if we remember when we did hormone signaling, there are hormone receptors in the cell, and when they bind the hormone, their shape changes, and then they are able to go into the nucleus and bind DNA and affect transcription. Right? So this is another very common way that protein function, gene expression, sort of, can be affected by binding to other molecules and ions. And we looked at in the nervous system, we had ion channels, and the ion channels function was being affected by voltage, etc. So many other things. We'll just put other. Proteins are sensitive to many things, and all of those things can affect their function. Binding to other molecules and ions. I think there was some other things. Other things also, protein folding can be affected. Protein folding. I think things like disulfide bond formation could be very important. Okay, so all these processes are done in a regulated way. They're not just randomly happening. So as the cell is responding to its environment or responding to other signals, things are happening in the cell to control which of these things are done to a particular protein to get its function to change in the way that best suits the cell in that moment in time. Okay, so disulfide bonds could be formed to make the protein structure different. And big one, protein degradation. Right? So there's also, even after the protein is made, when its function is possibly no longer needed, then it undergoes protein degradation. And there are even processes in the cell that identify when a protein needs to be downregulated, and certain things will happen to ensure that those proteins are broken down back to their amino acids. I mean, amino acids got to come from somewhere. Broken down, okay, back to their amino acids, so that maybe other proteins more useful to the cell can be made. Okay, so all these things happen to the protein once it's been made. So we'll just call these, point number 5, we'll call these post-translational modifications. So after the translation process has happened, all these various things can happen to the protein to adjust its function. Okay, so I hope that has been useful. As we can see, the story of gene expression is not so simple, and lucky that it is not so simple, because then the more complexity that you add to the process, the more regulation you can have, and therefore the kind of more complex an organism you can build, right? So there we have it. We've got genes, right from the DNA being remodeled to make certain genes less and more accessible, to the regulation of how often transcription can happen, to how long mRNAs can hang around in the cytoplasm, to how often those mRNAs are used for translation, to even after the protein has been made, things are happening to the protein to adjust its function. All of these things affect how the gene information is being used to affect cell and organism function. All right, guys, I hope this has been useful. Any questions, do let me know. Any suggestions for videos that are needed or that you might find useful, again, do let me know. Good luck, guys.